for everyone, especially to our beloved lecturer, Madam Diana. Let me begin by asking you a simple question. Have you ever wondered how pharmaceutical companies ensure the safety, purities, and effectiveness of the medicines we take every day? The answer lies in a powerful analytical technique known as chromatography. Therefore, here we will present to you about the application of chromatography in pharmaceutical industry. So, what is chromatography? Chromatography is a laboratory technique used to separate, identify, and quantify the components of a mixture. It works based on the principle that different compounds in a mixture will move through a stationary phase at different rates when pushed by a mobile phase. This difference in movement allows scientists to isolate and analyze specific substances even in very complex mixtures.
And interestingly, chromatography gets its name from a technique first used in the late 19th century to separate pigments in a complex mixture. The term comes from the Greek words chroma, meaning color, and graphene, meaning to write, because the earliest experiments involved separating colored plant pigments on paper.
Now, why is chromatography important in the pharmaceutical industry? In pharmaceuticals, chromatography plays a critical role in every stage of drug development, from the discovery of new drug compounds to quality control of finished product. It is used to analyze the purity of drugs, detect impurities or degradation products, and ensure that drugs meet strict regulatory standards set by agencies like the FDA. Without chromatography, it would be nearly impossible to guarantee that the medication reaching patients are both safe and effective. To sum it up, chromatography is more than just a separation technique. It is the guardian of drug quality and safety in the pharmaceutical world. With that, let's explore deeper on how this technique is applied in the real pharmaceutical industry.
Hi everyone, I'll be covering the theoretical part of our topic, how chromatography works and why it is so important in pharmaceutical analysis. Let's start with the basic principle behind it. Chromatography works on the principle of differential partitioning between two phases, the stationary phase and the mobile phase. When a mixture moves through the system, each component interacts differently with these two phases. Some parts of the mixture bind more strongly to the stationary phase, while others are carried along more quickly by the mobile phase. This difference in interaction causes the components to separate. To break it down further, first, the stationary phase is usually a solid or a liquid that is supported on a solid surface, like silica gel or polymer beads. This phase stays fixed in place, usually inside a column or on a coated plate. Next, the mobile phase, which can be a liquid or gas, flows through or over the stationary phase, carrying the components of the sample with it. Each compound in the sample has a different level of affinity toward these two phases. A compound that interacts more with the stationary phase will move slower, while one that prefers the mobile phase will travel faster. Over time, this allows the different components in the mixture to separate into distinct zones, and that is how we are able to analyze them individually. In pharmaceutical applications, this principle is used to analyze and purify complex drug mixtures. Techniques like high-performance liquid chromatography, gas chromatography, and thin-layer chromatography are chosen based on the type of drug and the purpose of the analysis. This theoretical foundation makes chromatography extremely useful for identifying active pharmaceutical ingredients, detecting impurities, monitoring degradation, and ensuring batch-to-batch consistency. So how does this look in practice during pharmaceutical testing? First, a sample is injected into the chromatography system. This sample usually contains a mix of compounds, for example, a drug and its potential impurities. The mobile phase then carries the sample through the system. As it moves, the components start to separate depending on how they interact with the stationary phase. A detector at the end of the system reads the separated compounds and converts them into signals, which appear as peaks on a chromatogram. Each peak represents a compound, and the size or area of the peak can tell us how much of that compound is present. This process is used to check the identity, purity, and concentration of compounds in a pharmaceutical product. For different types of testing, different forms of chromatography are used. HPLC is common for non-volatile drug compounds. GC or gas chromatography is ideal for volatile solvents. And ion chromatography is used when we need to analyze charged particles, like counter ions in drug formulations. All these methods are essential for quality control, regulatory compliance, and ensuring that medicines are both safe and effective.
everyone, I'm Sumaya and I will explain the methodology how chromatography is used in the pharmaceutical industry. So now let's start with the question, what types of chromatography are used in pharma? There are four main types. First, paper chromatography, known as PC. It's a basic method used to separate small things like food dyes or amino acids. It's cheap and simple, but not very useful for big or complex drug testing. Second, thin layer chromatography, known as TLC. This uses a plate coated with silica. It's helped check the purity of a sample or find contaminants. It's better than PC, but still give only simple results. Third, column chromatography, CC. It uses a tube filled with a solid material. We pour the sample through it to separate or clean up the different parts. This is more useful for bigger or bulk samples. Last and most important is HPLC, which is high performance liquid chromatography. This is the main one used in the pharma industry. So now the next question is, how is HPLC used in pharma? HPLC is used to identify, measure, and check the purity of medicines. It's fast, very accurate, and can detect even tiny amounts of impurities. So, here's how it works. A small amount of the sample is injected into the HPLC machine. A liquid solvent pushes the sample through a column filled with a solid material. Inside the column, the different parts of the sample separate. Then, a detector measures each part and shows a graph called a chromatogram. Each peak on the graph shows one component of the drug. So, let's look at a simple example. Imagine we are testing paracetamol tablets. The graph will show a big peak for paracetamol. If there are any small peaks, those are impurities. If these are too high, the medicine fails quality control. The size of the paracetamol peak tells us if the tablet has the correct dose, for example, 500 mg. So in summary, chromatography has many types, but HPLC is the most used in pharma. It's helped make sure medicines are safe, pure, and effective before they reach patients.
Now that we have understood how chromatography works and why it is crucial, let's look at a real-world example of how it is applied during pharmaceutical quality control.
Gas chromatography detecting residual solvents. In pharmaceutical drug synthesis, organic solvents such as methanol, acetone, ethanol, or dichloromethane are often used as reaction media or for purification. However, trace amounts of these solvents can remain in the final product as residual solvents. Gas chromatography is important because residual solvents can be toxic to patients. Therefore, their levels must comply with regulatory guidelines, such as ICH-Q3C. ICH-Q3C is a guideline established by the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use that focuses on controlling residual solvents in pharmaceuticals, which sets permissible daily exposure limits for different solvents based on their toxicity class. As we know, gas chromatography is the standard technique to detect and quantify this residual solvent. The chromatogram shows distinct peaks at specific retention times corresponding to each solvent. Each peak area is proportional to its concentration in the sample. For example, methanol appears at 2 minutes, acetone appears at 2 minutes and 48 seconds, and dichloromethane appears at 2 minutes and 5 seconds.
But if the analysis shows that any solvent is present above the regulatory limit, the drug fails the quality control, which is QC process. This means the batch cannot be released for public use. It either needs to be reprocessed or discarded altogether. But if all solvent levels are within safe limit, the product is cleared and deemed safe for consumption.
In conclusion, chromatography is critical in pharmaceuticals because it can ensure that no harmful residual solvents or impurities remain, protects patient safety from toxic exposure, and meets strict regulatory compliance in international standards such as ICH-Q3C. This is necessary for product approval and marketing. Furthermore, the impact of without the help of chromatography can lead to undetected impurities or residual solvents could remain in medicines. Next, this could lead to serious health risks such as toxicity, organ damage, or adverse drug reactions. Additionally, it could cause regulatory rejection, product recalls, and damage to a company's reputation.